Hello, my name is Chandler Robinson. I am a junior biology major, chemistry minor here at Oglethorpe, and for my bio seminar talk today, I'll be discussing implications of novel SIV treatment for zoonotic disease, specifically HIV, the human immunodeficiency virus. Motivation for this topic came about as I was listening to a medical podcast in which they were discussing HIV, the mechanism and how it affects the body. Then they went on to talk about how there is currently no cure for HIV, only treatments. And in today's age, with such advancements in science and technology, this just was very intriguing on how something like HIV, similar to that of cancer, is not able to be cured. Also, with the current COVID pandemic, COVID and HIV are both zoonotic diseases. So this is also a very interesting correlation to make and providing better understanding to both of these zoonotic diseases. So as far as what I'll be discussing today, I'll be talking about what is zoonotic disease, transmission from animals to humans. Then I'll move on to how it affects the immune system, and as well as how it integrates virally into the host genome. And then the research in this presentation is discussing the editing of SIV, the simian immunodeficiency virus, in which they use CRISPR to cut out specific regions of the genome, both ex vivo and in vivo, along with in vitro testing. And then they provide the results and conclusions of how it is both present and effective within the body. And then I'll give you some take-home points, mainly that it is a one-shot manner, along with my references. So to begin, what is zoonotic disease? Zoonotic disease, also referred to as zoonoses, are germs, in this case viruses, that transfer from animals to humans that can cause symptoms ranging from mild to severe. And to have a quote here to go along with that from the CDC stating, scientists estimate that more than 6 out of every 10 known infectious diseases in people can be spread from animals, and 3 out of every 4 new emerging diseases in people come from animals. I included this quote as it just shows the ramifications and the importance of understanding zoonotic disease that is very prevalent in our society, although we may not realize it. And the graphic on the right goes along with this by just showing how animals population and human populations continue to merge together. And along with this mergence, more and more zoonotic diseases are expected to grow with this in a linear fashion. So as the human population continues to encroach on the animals' habitats and environment, it is expected that the emergence of new zoonotic diseases will grow with that convergence. So how exactly does zoonotic disease, in this case HIV, transfer from animals to humans? So discussing transmission, specifically from the pan-troglodyte troglodyte, also referred to as the common chimpanzee from Central Africa, has been determined over years of research to be the origin of HIV in the form of SIV. So HIV began as SIV, the simian immunodeficiency virus, in the chimp, and then through one of 4 modes of transmission, or all 4, made its way to humans, which it became the human immunodeficiency virus. So these 4 modes of transmission, going from top to bottom, beginning with the image of a mosquito, is going to be vector-borne transmission, which is the transmission of these diseases, again HIV, through a carrier. So a mosquito, a tick, a mite, another simian species will come in contact with the infected monkey, the infected chimp, and then come in contact with humans. And this is a means of transmission. Below that is just showing a image of contact, which can be direct or indirect. Direct contact involves directly interacting with bodily fluids, so sweat, blood, saliva, feces of the infected chimp. That is another mode of transmission. And there is also indirect contact, which is just interacting with the environment in which that animal dwells. The bottom two is going to be waterborne transmission and foodborne transmission, in which both involve just consuming either water or food source that has been contaminated with the animal's feces, again, and other bodily fluids. So after transmission, what effects does HIV have on the body? HIV affects the immune system, in which it seeks to compromise the host immune system. It does so by targeting CD4 T cells. CD4 T cells are a major immune system cell, also referred to as helper cells, which are responsible for recognizing incoming infections or issues within the body, binding to that, and then recruiting other cells, being CD8 cytotoxic cells, phagocytes, or antibodies, to come and eliminate the infection. So HIV compromises these cells by integrating in and causing some degradation. So on the left, this is shown. On the far left, the brighter blue image is a healthy, full CD4 cell that is active within the body. As you can see, there are no holes and it is very full. While on the right image is a CD4 cell that has been infected with HIV. As you can see, it is very broken down and degraded. There's a bunch of holes and gaps within its structure. And then there's also these little yellow specks everywhere. These yellow specks are the virons, so the actual HIV virus that has replicated within the cell and now is budding. So the infected cell on the right will either undergo apoptosis, which is programmed cell death, or just lyse open. So this cell will die and then release these virons, these viruses, to other cells. And so this is how it travels throughout the body to infect all these cells. So as CD4 cells continue to die and are eliminated due to this infection, the immune system becomes compromised. And at progressive stages of HIV is referred to as AIDS, which is the acquired immunodeficiency syndrome, in which the CD4 count in the body has become so low the host immune system is no longer able to effectively fight off infections, externally and internally. These internal infections arise from OI, which are opportunistic infections, which are those that exist in the body currently, but are constantly being suppressed by the immune system. And now that the immune system is compromised, these opportunistic infections are allowed to arise and propagate and cause issues within the body. So how exactly does HIV integrate into these CD4 cells? It does so through viral integration. So what is this process of viral integration? To first give you some background, HIV is a retrovirus, which just means it has an RNA genome in which when it enters the cell, the RNA reverse transcribes to DNA, and this DNA is then able to integrate into the host DNA, the DNA of the cell. So how this works is displayed on the left, in which we have an HIV virus, the human immunodeficiency virus here, and then we have a CD4 cell. So HIV undergoes a process of docking, which it binds to receptors on the CD4 cell, and then fuses through the membrane into the cell. Once in the cell, the viral RNA is released, reverse transcribed into cDNA, and then turns into double-stranded DNA. And then the viral DNA can now integrate into the chromosomal DNA, which is just the DNA of the cell, through a process of integration. Now that it has integrated into the host genome, this cell is going to use its replication machinery to replicate this virus, and then it will lyse or undergo apoptosis and go on to infect other cells in the same fashion. This can occur in both active and non-active CD4 cells. Active meaning a CD4 cell that is fighting off infection and functioning in the body properly to increase immunity, while non-active cells are those that are dormant and are just waiting to be recruited to fight off infection. So since HIV is able to integrate into both active and non-active cells, this gives great issue to the current treatment, which is ART, referred to as antiretroviral therapy. Antiretroviral therapy acts by a regimen of drugs which is taken over weeks, periods, or periodically to reduce the viral load in the body, so the presence of the virus in the body, but can only do so by targeting the cells that are active, meaning these CD4 cells that are active and have HIV are the only cells that can be eliminated by ART. Those non-active CD4 T cells are unaffected by ART. So PWH, people with HIV, will receive this treatment, reduce the viral load of these active cells. However, weeks, days, months later, these non-active cells then become active, allowing HIV to again spring up, known as viral rebound, and continue to replicate and propagate within the body. So this issue with antiretroviral therapy, being unable to cure HIV, is the basis of the research I'll be discussing in this presentation, in which they seek to edit SIV. The simian immunodeficiency virus is going to be used as a model to better treat HIV in the future. So in 2020, publication of Nature, after 5 years of research, they have come to a method of using CRISPR-based editing of SIV proviral DNA in ART-treated non-human primates. So CRISPR is just a genome editing tool of the simian immunodeficiency virus targeting the proviral DNA, and taken in parallel with antiretroviral therapy of non-human primates being the rhesus monkey. So the basis of this research again is they recognize the shortcomings of ART, although acknowledging that it is doing a decent job of mitigating some symptoms, but they sought out a better method in which they arrived at AAV9, CRISPR-Cas9, also referred to as CRISPR-SA-Cas9. SA just standing for Staphylococcus aureus, the bacteria which it was derived from. So AAV9 is the adeno-associated virus number 9, which is a virus in which they remove the viral DNA and replace with their own programmed DNA. AAV9 is going to act as a vehicle to deliver CRISPR-Cas9 to all tissues of the body. They came about using this method as in prior studies in mice and rats. AAV9 has been shown to deliver to all tissues of the body successfully. So that's why they're using this here. And they hypothesize that if AAV9, CRISPR-Cas9, can properly target and remove large segments of proviral DNA, then the chance that the virus will replicate competently is low or mute. So by delivering this CRISPR-Cas9 to these infected cells, the chances that these cells replicate is very low or zero. So first, what exactly is CRISPR? So CRISPR-Cas9 foundationally has been approved this year in 2021 by the FDA to treat sickle cell anemia, which is a mutation in red blood cells that can cause a variety of issues in those that have it. And it has also been used to look at editing the genome of newborns and babies to limit these mutations and increase the quality of life. So there's a foundational aspect to this as they were looking for new treatment options. CRISPR seemed to be a good method to do so, as it has been proven to work well in diseases of similar interest. So how does CRISPR work, and what is CRISPR? CRISPR stands for clustered regularly interspaced palindromic repeats. That is besides the point. The two main aspects of CRISPR are gRNA and the Cas9 enzyme. So to explain these, on the right I have a graphic, which is a very simplified version of how CRISPR-Cas9 functions. So to begin, Cas9 is an enzyme, more specifically an endonuclease, which is responsible for cutting targeted portions of the DNA out, in which they refer to it as molecular scissors, while gRNAs are guide RNAs, which are programmable sequences of RNA to target specific regions of the DNA that the edit they want to happen to occur. So by combining Cas9 and gRNAs together, you insert this into the body, and then it targets this specific sequence of DNA, taking it into the Cas9 enzyme, shown in step 4. The DNA is unwound, the gRNA targets this region, and then Cas9 can come in and excise or cut out this targeted region. In step 5, after excision, the DNA is then able to repair or be disabled. In this experiment specifically, they are looking to disable targeted regions of the SIV genome to render it incapable of replicating. These targeted regions, shown in the bottom left, labeled figure A, were rendered on a computer system. So these are the guide RNAs that were programmed to target the specific regions of the SIV genome, which are the 5' LTR gRNA, the GAG gRNA, and the 3' LTR gRNA. So these are the three targeted regions that if you can excise one or both, the chances of replication of SIV is thought or hypothesized to be 0. So after identifying their targeted areas, how exactly do they go about testing this? In which they began in vitro, or out of monkey, with cell cultures. So in this top graphic here, they acquired cell cultures as a proof of principle technique and method to assure that CRISPR is actually targeting and excising the regions they want and works in theory, and not just in theory. So they acquired these cell cultures. They gave these cell cultures SIV MAC239, which is just the simian immunodeficiency virus found within rhesus monkeys, leading to these infected cells. So these cell cultures receive SIV. These are now infected cells. And then they treated these cells with the AAV9 CRISPR-Cas9, in which they would expect to see cells with these excised regions. And to show this, they ran a gel. So shown here in D, it's just showing that their targeting was successful. So as a control, they have some untransduced cells, which show no excision. And then they have the cells treated with CRISPR, in which they show the full link to the genome, and then the actual excised regions done by CRISPR. So these excised regions show that it was successful in removing these targeted areas. So following success here, they moved into in vivo and ex vivo studies. So in the monkey, or in vivo testing, began by acquiring 4 rhesus monkeys, assuring that these monkeys had no antibodies to AAV9 and had not been previously infected with SIV. And then they took all 4 monkeys and they intravenously gave them SIV MAC239. 2.5 months post-infection, all 4 monkeys began to receive ART treatment, so antiretroviral treatment. Following this, 5 months post-infection, 3 of the 4 monkeys received CRISPR intravenously, so in a one-shot method they received the AAV9 CRISPR-Cas9, and then 1 of the monkeys did not receive CRISPR but continued to receive ART. So throughout the entirety of the experiment, all 4 monkeys received ART, but only 3 of the 4 received CRISPR, while the 4th remains as a control to assure that CRISPR actually works. And then 7 months post-infection, these monkeys had blood samples taken from peripheral mononuclear blood cells, which are the main blood cells of the immune system. So in vivo, they removed these blood cells to examine them ex vivo, which is shown here in this figure, in these gels. So beginning at the top, again, they are looking at 2 infected monkeys, so KM77 and KP43 are 2 monkeys that received CRISPR treatment, while KK09 only received ART and no CRISPR treatment. So all 3 monkeys have SIV, only 2 received CRISPR. This is important to note as these targeted regions, these LTR and GAG regions, were successfully excised in vivo in these monkeys. So these are the excised regions shown here, while KK09 that received no CRISPR as a control shows no excisions happening. And then at the bottom, they are just testing to make sure the presence of SA-Cas9 within these monkeys is present, along with, again, looking at these excised regions. So you would expect to see the excised regions as well as the presence of Cas9 in the 2 monkeys that received CRISPR and none in the monkey that did not receive CRISPR. So this is 7 months post-infection. Following this, these monkeys were necropsied and examined more thoroughly. So after necropsying these monkeys, which is humanely killing them and then examining all parts of their body, so kidney, spleen, liver, heart, all these different areas were examined for both the presence and effectiveness of AAV9 CRISPR-Cas9. So shown on the left here in labeled figure A and B in this vertical column, they are testing for the presence and copy number of SA-Cas9 in a variety of organs throughout the body, in which there was a very high presence of SA-Cas9 in the spleen and liver, as well as the lymph node. The lymph node is known to be a major viral reservoir, so a high presence of viral load, which is important to note. And then in B, they were testing for actual expression. So although it is present in these regions, is it actively expressing? In which the 2 important things to note is the bone marrow and kidney showed no expression in those regions, in which they alluded to further testing is needed to understand why. In this figure on the right, labeled A, they show both a graph and a gel, specifically of the lymph node, as again it is a major viral reservoir, so they wanted to test the effectiveness here. So we're looking at the graph. This is the percent of proviral DNA in the lymph node prior to treatment and after treatment, in which the important things to note are going to be the KK09 is the untreated monkey that received ART but no CRISPR, saw a 20% reduction in viral load, but while the CRISPRs that received both ART and CRISPR in those monkeys saw as much of a 95%. So the peak is a 95% reduction in viral DNA in the lymph node, which is displayed here on the right in a gel, which shows the various parts of the lymph node and the targeted regions that were excised in both the monkey that received CRISPR and ART, which is KM77 and KP43, and then comparing that to the control monkey that only received the antiretroviral therapy. So they concluded that due to a small sample size of only 4 rhesus monkeys, that further testing is needed, as well as a longer duration would prove to show better results. This is promising, as it is the first and novel method of treating SIV using CRISPR to have ever been performed, and they express that they are happy with the results and the outcome of this experiment as they move forward. So not only what to take away from this research, but what to take away from this presentation. By expanding our knowledge in this particular area is going to be crucial to prevent future zoonotic disease as humans and animals continue to grow more intertwined. So this research is crucial as more and more zoonotic diseases continue to spring up. The treatment of SIV is going to have major implications on treating HIV as well as other diseases that are yet to come and that have yet to be cured. AAV CRISPR-Cas9 is also, again, the first treatment of its kind and is a promising treatment for PWH, people with HIV, and other diseases in a one-shot manner. This idea of a one-shot system, so instead of receiving constant treatment like ART or receiving constant treatment like chemo, you are able to receive a one-time shot that can actively work within the body and provide these corrections and cure HIV, sickle cell anemia, and eventually they hypothesize cancer. So this research is very new and is going to have to be continued to be tested, but it is very promising for those with HIV, AIDS, and other diseases that continue to plague this world. These are my references. I would like to thank you for paying attention and giving me your time. I hope you learned something valuable here. And if you have any questions, feel free to leave them below or shoot me an email. Thank you. Have a nice day.